Imagine you're trying to manage traffic in this massive, just sprawling metropolis.
Yeah. Yeah.
But the only tool you actually have to do your job is a static black and white photograph of the city skyline. And it was taken from a helicopter at, like, noon.
So you can see the buildings, basically.
Right, exactly. You see the big skyscrapers, you get the general shape of downtown, but you have zero visibility into the actual traffic patterns. You can't see the power grid, and you certainly don't know, what emails are being sent inside those office buildings to actually coordinate the city's operations.
Yeah, which is kind of essential if you're running a city.
Exactly. And for decades, traditional bone marrow biopsies have functioned a lot like that static photograph. You get this broad structural snapshot of a patient's cellular landscape. But you're largely blind to the underlying functional networks that are actually driving the disease.
Yeah, that's a perfect way to put it, actually.
Yeah. Welcome to the Digital Pathology Podcast for today's deep dive. For all the trailblazers joining us today, so, the pathologists, the oncologists, the hematology specialists out there, we are doing a journal club exploration of a paper that essentially takes that static photo and upgrades it into a live, interactive, predictive 3D map.
Right, and the paper we're looking at today is titled Advancements in Bone Marrow Biopsy, the Role of Omics and Artificial Intelligence in Hematologic Diagnostics. It was published by Maryam Alwahaibi and Nasar Alwahaibi, and this was in Frontiers in Medicine back in March of 2026.
I have it right here.
Yeah, and what this work really synthesizes for us and why it matters so much to the trailblazers listening is this stark reality that our traditional methods for assessing bone marrow are just hitting a wall. I mean, these biopsies are the absolute cornerstone for diagnosing leukemias, lymphomas, marrow failure conditions, but they're hitting a very hard biological and, honestly, operational ceiling.
Yeah, and I feel like before we get into the shiny new tech, we have to start by looking at how that old system is breaking down, right? Because that really sets the stakes for everything else.
Absolutely.
I mean, the gold standard has always been these conventional modalities, right? Histomorphology, flow cytometry, cytogenetics. A pathologist takes a bone marrow sample, uses an H&E stain, which, by the way, hematoxylin and eosin, that dye technology is over 150 years old.
Yeah, it's literally 19th century technology.
Right. It turns nucleo blue and cytoplasm pink, and then you just look down a microscope.
And that visual assessment is just incredibly fraught because, you're dealing with highly overlapping morphological features. The visual differences between a reactive state, so say a patient's marrow is just responding to a severe infection, and early malignant changes, like a developing myelodysplastic syndrome.
They look almost identical sometimes.
Exactly. The differences can be incredibly subtle. And because the entire interpretation relies on a human brain processing those subtle visual cues, the process is just inherently subjective. I mean, the medical literature is absolutely packed with data showing really high rates of intra-observer and intra-observer variability.
Wait, meaning two different doctors or even the same doctor on different days?
Both. You can hand the exact same slide to 3 highly trained, board-certified hematopathologists and literally get 3 slightly different interpretations of the blast count or, the degree of dysplasia.
Or, like, a pathologist might look at a slide on a Tuesday morning after their first cup of coffee, right?
Right.
And then they look at that exact same slide on a Friday afternoon when they're experiencing severe cognitive fatigue, and they just grade it completely differently.
Right. And plus, scanning these slides manually is incredibly slow.
Yeah.
So when you combine that slow subjective process with the current global shortage of expert bone pathologists, you just get massive clinical bottlenecks.
Which is terrible for patient care.
It is. And it's especially disastrous for rare or, dedifferentiated tumors that just don't play by the standard visual rules anyway.
Okay, but let me push back on something here, just playing devil's advocate for a second.
Sure.
We have relied on microscopes and trained human eyes for decades, right? And we've cured or managed countless patients. Why is this methodology suddenly insufficient right now? Like, is the underlying biology of these diseases actually getting more complex? Or are our standards for precision medicine just demanding way more than the human eye can deliver?
That's a great question. And what's fascinating here is that the underlying biology of a leukemia cell hasn't actually changed over the last 50 years.
Okay.
But our understanding of its molecular heterogeneity has absolutely exploded. We now know that 2 leukemias that look morphologically identical under a microscope down to the exact same shape, size, and staining pattern, they might actually be driven by completely different genetic mutations.
Oh, wow. So they look the same but act totally differently.
Exactly. And more importantly, they might require completely different targeted therapies to achieve remission. So a human expert simply cannot manually quantify those high-dimensional molecular complexities. I mean, the human eye cannot see a genetic mutation, right? The human eye can't see a misfolded protein cascade.
Right. If the human eye maxes out at the cellular level, then we have to drop down to the molecular level to see what's actually happening behind the scenes. And I think this is where the authors bring in the whole multi-omics, multiverse, as they call it.
Yeah, the multi-omics approach is key here.
Because to get past the bottleneck of that subjective visual analysis, the field had to develop technologies that interrogate the marrow at a microscopic, really functional level.
Yeah, and these omics technologies, they systematically break down the marrow ecosystem layer by layer. The foundation, of course, is genomics and transcriptomics. We're moving far beyond the era of just sequencing a basic DNA blueprint. We are actively hunting for specific clonal mutations, like single nucleotide polymorphisms, or SNPs, that tell us exactly where the DNA code broke.
And then transcriptomics comes in.
Right. With transcriptomics, we're mapping out RNA splice variants. We can look at the active inflammatory signaling pathways to see not just what genetic code is broken, but what the cell is actually doing with that broken code in real time.
And the paper delves into epigenomics too, which is where I think the mechanics get super fascinating.
Yeah.
Because we aren't just looking at the code itself anymore, we are looking at DNA methylation and chromatin remodeling.
Yeah, the switches.
Exactly. They function as these physical roadblocks or access points on the DNA. There's the switches controlling lineage specification, determining whether a hematopoietic stem cell turns into a red blood cell, a platelet, or a white blood cell. When a methyl group attaches to the wrong spot and flips the wrong switch, it can completely silence a tumor suppressor gene, right? Which just kicks off malignant transformation without actually altering the underlying DNA sequence at all.
That's spot on. And beyond the genome and the epigenome, we get into what the authors highlight as the emerging frontiers. Lipidomics is a prime example of this.
Which is crazy to me because lipids are just fat, right?
Yeah. Well, historically, yeah. Lipids were viewed primarily as just passive structural components of cell membranes, or like inert energy storage.
Mm.
But lipidomics reveals that specific lipid species, things like ceramides or sphingolipids, they actively regulate stromal hematopoietic communication.
Wait, really? They're sending signals?
Yes. They form these specialized rafts on the cell membrane that aggregate signaling receptors. By changing the lipid composition, they physically remodel how the marrow microenvironment allows mesenchymal progenitors, osteoblasts, and immune cells to communicate with each other. They're essentially rewriting the entire communication network of the marrow.
That is wild. And speaking of wild communication networks, the section on microbiomics and the gut-marrow axis completely shifts how we have to think about localized disease.
Oh, absolutely.
The paper details how systemic immune function and gut dysbiosis, so severe imbalances in your gut bacteria, how they indirectly impact marrow homeostasis, and even how a patient responds to chemotherapy. It operates a lot like a complex supply chain, Going back to our city analogy, you can have a local factory, the bone marrow, that is fully staffed and ready to produce blood cells. But if there is a massive traffic jam, 3 towns overrepresenting inflammation in the gut microbiome, that traffic jam delays the raw materials and sends stressed, agitated drivers into your local town.
I love that analogy. It's exactly like that. The systemic inflammation driven by the gut microbiome alters the cytokine balance that's circulating in the blood, which eventually, of course, hits the marrow. And the marrow is highly sensitive to inflammatory cytokines like interleukin-6 or TNF-alpha.
Right.
Chronic exposure to those signals can push hematopoietic stem cells to exhaust themselves or bias them toward producing myeloid cells over lymphoid cells. And for the trailblazer listening and trying to apply this to their practice, these omics layers aren't just esoteric biology, right? They fundamentally refine our diagnostic classifications. They detect targetable therapeutic vulnerabilities and minimal residual disease at levels that a traditional H&E stain would completely miss.
But we run into a serious computational wall here, though, because omics provides an absolute mountain of molecular data. But a pathologist still needs a way to interpret the physical tissue morphology. Like, we need a bridge between the digital data of a sequenced genome and the physical reality of the tissue on the slide, without getting trapped in those subjective human bottlenecks we established earlier.
Enter artificial intelligence. AI acts as that bridge, serving as basically the ultimate analytical lab partner.
Okay, let's break that down. How does it apply to histology, meaning the actual solid tissue biopsies?
Well, in conditions like myeloproliferative neoplasms, MPNs, one of the most critical diagnostic and prognostic tasks is grading the degree of bone marrow fibrosis. Traditionally, this is a highly subjective scoring system from 0 to 3 based on how thick the reticulin fibers look under the microscope.
Which goes back to the human error problem.
Exactly. But AI models have now been trained to evaluate those reticulin changes pixel by pixel, generating a continuous index of fibrosis, or CIF. So instead of a clunky 0, 1, 2, or 3, you get a highly granular, continuous metric. And this objective grading has been shown to predict disease progression in MPNs far more accurately than human estimation.
And the paper highlights specific automated tools too, right? Like MarrowQuant. Because when a human estimates cellularity, they usually just scan the slide and guess, saying something like, wow, looks like maybe 40% cellularity.
Yeah, it's a very educated guess, but still a guess.
Right. But MarrowQuant automates the mapping of adipocytes, bone trabeculae, and vascular structures. It systematically analyzes the entire slide, mathematically subtracting the area taken up by fat and bone to standardize the cellularity assessment. It just removes the human guesswork completely.
It does. And from solid histology, the authors moved to cytology, so the liquid bone marrow aspirate smears. Figure 2 in the paper outlines this comprehensive end-to-end clinical workflow. And what really stands out there is how the AI doesn't just analyze cells, it actively manages quality control.
Yes. The moment a slide is digitized, the first thing the AI does is run a QC protocol, right?
Right.
It aggressively rejects crushed artifacts, excessively thick areas of the smear, and severe staining outliers, which is huge because if a human technician prepares a poor smear, a human pathologist might waste 20 minutes squinting at deformed cells just trying to force a diagnosis. The AI instantly flags the preparation as non-diagnostic.
Exactly. And if the slide does clear QC, the system moves to cell detection, segmentation, and classification, generating an automated differential histogram of cell types.
Oh.
It flags suspicious morphological patterns and routes those specific regions of interest directly to the human expert for review. It functions as a highly sophisticated triage system. And the AI possesses diagnostic capabilities that really push the boundaries of current pathology. The paper discusses models like dysplasia net. In myelodysplastic syndromes, identifying hypogranulated dysplastic neutrophils is clinically vital, but notoriously difficult.
Why is it so hard?
Because a normal neutrophil has a cytoplasm packed with specific granules. In MDS, those granules can be sparse or absent, but the visual difference is incredibly subtle, especially if the slide stain is even slightly washed out. DysplasiaNet detects this specific morphologic deficit objectively and consistently, standardizing a task that routinely trips up human observers.
And it plays a massive role in acute promyelocytic leukemia, or APL, too, because APL is a true hematologic medical emergency. Patients can rapidly develop disseminated intravascular coagulation. I mean, they can essentially bleed to death before a molecular test even comes back from the lab confirming the diagnosis.
It's terrifyingly fast.
Yeah. And APL is driven by a specific genetic swap, right? The T1517 translocation. The AI can rapidly recognize the morphologic patterns associated with this translocation, like hypergranular cells with folded nuclei or bundles of Auer rods, in minutes. It reliably distinguishes APL from other acute myeloid leukemias just by analyzing the geometry and texture of the cells, which allows clinicians to start life-saving all-trans retinoic acid therapy immediately.
Right, and if we connect this to the bigger picture, the algorithm is predicting genotype from phenotype. This is the paradigm shift. Deep learning models are linking the visual shape, texture, and spatial arrangement of cells directly to underlying genetic mutations.
That feels like magic.
It really does. A neural network processes pixel intensity gradients and high-dimensional spatial relationships that our human visual cortex literally did not evolve to recognize. The paper notes AI can identify subvisual histologic features strongly associated with TET2 mutations or del5q MDS. It can look at the shape of a cell and say, with high statistical probability, this cell is missing the long arm of chromosome 5.
Okay, let me stop you there, because predicting genetic mutations from a simple slide scan sounds almost too good to be true. If AI can do this rapidly and cheaply, why isn't every community hospital and clinic on Earth running this software today? Like, does integrating AI actually save time, or does it just create a new operational nightmare where pathologists spend all day babysitting algorithms, verifying these black box outputs, and fighting with IT systems?
The friction points you're bringing up are exactly why this isn't universally adopted yet. The black box nature of deep learning is a massive regulatory and clinical hurdle. A convolutional neural network might correctly identify a TET2 mutation, but it outputs a probability score without explaining its biological reasoning.
Right. It just says, trust me.
Yeah, and in medicine, you cannot simply tell a patient they have a specific leukemia because a proprietary algorithm said so. Pathologists require an auditable trail of evidence to sign off on a diagnosis, and black box models just don't provide that.
There's also a severe lack of generalizability, right? AI models are incredibly brittle when taken out of their training environments. You could have an algorithm trained on hundreds of thousands of pristine slides from a well-funded, high-end academic center, but it might completely collapse when you deploy it in a rural community hospital.
Oh, totally.
Yeah.
Because the scanner resolution is different, or the glass slides are a millimeter thicker, or the eosin stain has a slightly different pH causing a minor color shift.
Right. And the omics side has equally daunting hurdles, doesn't it?
Definitely. High-throughput sequencing assays are expensive, technically complex, and demand massive bioinformatics infrastructure. Furthermore, a bone marrow biopsy is a physical procedure. If you get a dry tap or a hypocellular sample, extracting enough high-quality RNA or lipid material for multi-omics analysis is incredibly difficult. A low-quality sample inputted into a high-end omics pipeline will just output biased, incomplete molecular profiles.
And then you have the absolute headache of data integration. Because genomic data is static, your DNA mutations are relatively fixed. But transcriptomic or metabolic data is highly dynamic. It fluctuates based on the time of day, the patient's stress levels, or recent medications.
Yes, exactly.
So mashing static structural data together with temporal functional data, and then trying to map that onto an AI-generated spatial map of the tissue. I mean, without established universal interpretation frameworks, it's a recipe for clinical paralysis. Are we looking at a bifurcated future where only elite, well-funded academic centers can offer this level of precision diagnostics, leaving community clinics relying on traditional microscopes?
The field recognizes that risk, absolutely,
Yeah.
and is actively engineering solutions to democratize the technology. To solve the AI generalizability problem, researchers are shifting toward federated learning.
Oh, federated learning. That's where you train the model without moving the data, right?
Exactly. Instead of a community hospital sending its sensitive, raw patient data to a central server to train an algorithm, which triggers massive data privacy and bandwidth issues, federated learning sends the algorithm to the hospital.
Ah, okay.
The model trains locally on the community hospital's uniquely stained slides, updates its internal mathematical weights, and then sends only those updated weights back to the central server. So that the global model learns from the diverse, messy reality of thousands of different labs without ever centralizing the raw data.
That's brilliant. And to solve the black box problem, the industry is forcing a shift toward explainable AI, right? These are systems architected to basically show their work. Instead of just spitting out a diagnosis of MDS with 90% confidence, explainable AI generates a heat map overlay on the slide.
Yeah, the heat maps are game changers.
It highlights the exact morphologic features, the specific hypogranulated neutrophils or the abnormally clustered megakaryocytes that actually drove the algorithm's prediction. It turns the AI from a mysterious oracle into a transparent assistant that the pathologist can verify and trust.
Right. And all of this development points toward the ultimate future vision outlined by the Alwahaibis in the paper, end-to-end computational pathology pipelines. The authors point to spatial multi-omic atlases, specifically mentioning multiplexed imaging techniques like Codex.
Code detection by indexing?
Yes, Codex. It uses DNA barcoded antibodies to visualize dozens of different proteins on a single tissue section simultaneously, without destroying the sample. It allows researchers to physically map genetic and proteomic alterations to their exact topographical niches in the marrow microenvironment.
So you can actually see that the cells with the TET2 mutation are physically clustering around specific blood vessels, while the healthy cells are pushed to the periphery. You get the 3D map with the traffic patterns and the electrical grid fully illuminated, going back to our very first analogy.
Exactly. It all comes full circle. To distill everything we've covered today for you, traditional bone marrow biopsies, relying on visual inspection of H&E stains, are foundational but inherently limited by human subjectivity and visual constraints. Multi-omics technologies, from the genomic code down to the lipidomic signaling rafts and the gut microbiome's systemic influence, reveal the invisible molecular machinery driving hematologic diseases.
And artificial intelligence is the engine that actually processes this overwhelming amount of data. It objectively quantifies tissue fibrosis, automates quality control on aspirate smears, and performs the highly complex mathematical translation required to link a cell's physical phenotype directly back to its underlying genomic mutations.
It is a complete paradigm shift in how we understand tissue.
It really is.
Which leaves us with a compelling philosophical question for you to consider as we wrap up. If artificial intelligence can eventually and accurately predict a patient's underlying molecular mutations purely by analyzing the visual shape, texture, and spatial arrangement of cells on a standard, inexpensive H&E slide, does the traditional boundary between a morphologist and a molecular geneticist even exist in the future? As you navigate this evolving landscape as a trailblazer, will your value be defined by the physical tests you order or by your expertise in curating the digital algorithms that interpret the tissue?
Man, that is a brilliant thread to pull on as we close out. From a static photograph of a city skyline to a living, interactive, predictive map of the human microenvironment. Thank you to all you trailblazers for exploring this paper with us today on the Digital Pathology podcast. We will see you next time.